now that we're in this room, we can, get our stuff all set up for our internal assessments, your individual experiments that you need to run. So, like right now, still have organizing to do because ideally a lot of you will be set up over there on those growing racks and other places, and get you a spot to get your materials all collected and everything. So we need to. I posted a new IA, data, how you're going to analyze your quantitative data that you build during your lab experimentation period. So we got to get back in that IA mode again. So tell me what the difference is between all those variables: controlled, dependent, and independent. Because it's confusing, right? And we can't ever talk enough about what the difference is, so we really get a good grasp on that. And option D, what we're learning right now, I posted some new videos last night and some quizzes. What is the difference between systolic and diastolic blood pressure? Because when you get a blood pressure, is it just one pressure? No, it's two, right? So it's one number on top of the other. So which number's on top, which number's on bottom, and what do they mean? Okay? So just whatever you can think of, and that graphic image is a guide. It kind of helps, explain it. It might be confusing, but maybe you can use that to help you answer that question too. And if you see, I'm trying to find a clipboard with a paper with all your IA information on it, and I saw it somewhere. I saw it like 20 times in the last day, but now when I want it, I can't find it. So if you see a clipboard sitting somewhere, oh, it's right. See, when you ask a question, you answer it when you ask for help. So I'm always impressed by that maxim. Yeah, like just asking a question instead of trying to figure it out yourself, and ask a question and then it's answered for you. Isn't that amazing? Okay, so work on that. It is cold out. Oh my God. I don't get cold. I'm cold today. No, not yet. Let's see. away from you there. Oh yeah, you're welcome. Oh, just make a guess, Bree. Right. Softball starting soon, huh? Yeah, you're gonna be out there. Games. Oh, it's got lead in there. Okay. Make a guess there, making the guess. Beating, pressure, resting. Oh. Oh. We got Alondra. So let's talk about variables, Alondra. Independent. How would you explain an independent variable? Ooh, it stands on its own. Okay, yeah, that's what independent means, right? Standing on your own. It's me. I can do it. Which is good, right? Sometimes you need a little help, though, right? Right? Okay, well, those colloquial explanations don't do the best job explaining in an experiment you're going to do what it means. Independent variable of your experiment, Esme. Well, I'll put that. We gotta learn this, Esme. We gotta know. Any other thing you could add to what an independent variable is? Besides standing on its own, strong, doing its own thing. What's that? What about manipulation? Okay, so the independent variable is that variable that you are manipulating. That's another word for it, the manipulated variable. So it's the variable that you change, that you're controlling, you're moving around, you're changing the levels for to see its effect on something else. Okay? So independent. So you change, manipulate, adjust. You're kind of in control of this one, okay? Like measuring the acidity, how different levels of acidity affects the growth of radish seeds. Your independent variable would be, the acidity, and you're going to have different levels of acidity, right? pH 2, 3, 4, 5, 6. You're controlling that pH, right? What about dependent variable? So dependent, what does that really mean in this context? Madeline. Okay, yeah, what you're measuring. Okay, what you're measuring, what that independent variable might affect. So in my example just now, with the plants and the pH, what would be your dependent variable? Yeah. What about the plants? You were kind of wondering how different pH levels would affect the growth of your radish plants. So your dependent variable might be, what might you be measuring about those plants? Well, you might want to measure the pH, measure that as a part of your independent variable. How what changes? The plants what? So measuring those plants, seeing how high they are. You can measure other things like the mass of the plants, the color. Maybe if you had a way to measure how much photosynthesis on a biomolecular level, you could measure that. But easiest way for us to measure how well photosynthesis is happening by measuring how tall they get or how big they get. So dependent is what you measure. Okay, what you're measuring. Okay, trying to figure out what that independent variable, its effect on that dependent variable. And the control variables, we can't forget about those. Aries Beth, what are the control variables all about? Okay, yeah. So something we don't want to change our conditions of our experiment. I like that term, like the conditions, the general conditions of our experiment. We want the same for all groups. So let's write that over here, controlled. We'll say in general stay the same. General conditions of experiment. Like in the example I'm just using about my plants and pH. We change the pH that we're growing our radishes in, but what do we want to keep the same for everybody? Yeah, like the radishes themselves may be the same kind of radish, the same age. We shouldn't get seeds from 2010 and compare seeds from 2023, right? So our seeds should be the same age, the same variety, the same amount, okay, of seeds we're using. What else should be the same besides the seeds? Temperature, yeah. What the temperature conditions we're growing all of our seeds in should be the same because that can affect growth. What else can affect growth? What's that? The sun? Okay, the sunlight, right? These should all have similar amount of artificial or actual sunlight. What else? Amount of water, right? And that's what's really nice about those Lee Valley seed starter trays, those styrofoam things that I showed you guys before. Those of you that are growing, those make sure that the amount of water is the same for all of your groups. And what do plants grow in? Soil. So the soil should be the same, right? The amount, the kind of soil. Tons of variables, right? So all of those things, how you measure them. Should you use different rulers for different groups? Absolutely not. You should not do that. Same ruler, same way, same method. You should measure on the same day, the same time. Measure in the morning and then measure later in the afternoon. Don't do that. You want to measure consistently all groups at the same time, the same way, the same ruler, the same. Okay? So, yeah. All those other conditions. The only thing that should be changing in this theoretical experiment should be what? The only thing that should be different between the groups should be the pH level, right? And if you can do that, any difference you see in the groups should be because of the pH, right? If other things are different, then who knows what's causing the difference. That's the power of the controlled experiment, the power of empiricism, the power of science itself. Okay, being able to get those variables dialed in. Now, is it possible to 100% control everything at all times? No, it's very, very difficult. That's why in science, having large numbers of replicants is important, and not just 5 or 10, but 30, 300, 3,000. The more replicants, the more trials, the more those variables even out. Like when they test drugs, could a drug work fine on 10 people? Yeah. But what happens when you start to give a drug to more and more people? Do you find more and more problems? You can. You know, different people, some people react to it. Could be rare, but you got to be aware of that. That's why they want to test on lots and lots of people. So you can't ever get the variables completely dialed in. There's always something you're not seeing, but you got to be aware of it. You got to try, and that's the point of this experiment. And try to get as many replicants as you can. All right. All right. Ooh, difference between systolic and diastolic blood pressure. Marcos, what do you think? What did you write down?
It could be the difference in blood, like how much is being input.
Oh.
Systolic, because like the heart is shaped more like narrow, I guess.
Okay. So the narrowness of the heart.
No, because like I guess like the pressure will like have it more compressed, like I guess.
Okay. Well, the heart compresses. That should raise or lower pressure. When the heart compresses, when it pumps. Okay. Esme? Or Jane? Yeah, in a way. Think about it when you have two different parts of the heart that contract, right? You got your top chambers and your bottom chambers. Your top chambers, these are the oh, wow. It's different. You got your atria up here, the top chambers, and you got your ventricles. So when the ventricles contract, they're just pumping blood into when the atria contract, you're just pumping blood into the ventricles. Oh, hello. It's me. I'm good. Yourself? Oh, Happy New Year to you too. Oh, check it out. All right. Appreciate it. All right. All right. See you. So your atria contract, it fills up those ventricles. That's one set of contractions. And then the ventricles contract. The blood pumps. But where does it go? Because we know that first atria, the blood's staying within the heart. But when the ventricles, when the bottom contracts, where's that blood going to go? Huh? Yeah, the lungs. And if it's in the left ventricle, it's going to go to the lungs. Man, this is really impossible now. Right here, this is going to go to your lungs. That's the right ventricle. But the left ventricle, this blood is going to go where? This is the aorta. And that's going to go to the body, all your body. Okay? So the ventricle's contracting. That's, the biggest, that's the most muscular, strongest contraction. The blood's going to go to your body. The atria contracting, that blood's just being pumped from one set of chambers to the other. So in terms of systolic and diastolic, systolic is when the blood's pumping out of the heart, okay, into either the blood, the lungs, or your body. And then the diastolic is when the atria are contracting and that blood is being pumped in the ventricles. And which one makes sense to be a higher blood pressure? When the ventricles contract or when the atria contract? Which one is a beefier set of chambers? Well, the atria are pretty small compared to the ventricles. And that diagram kind of makes them look a little more equal than they are. But your ventricles are a lot beefier, they're stronger, you know, it's a bigger, it's more muscle mass when it contracts. Well, the blue, that's showing the right side of your heart. Okay, the deoxygenated blood. So when the ventricles contract, this is the strongest portion, the strongest contraction you're going to have, you know, going to your body and your lungs. When the atria contract, that's less forceful. So if you look at your blood pressure readings, like the kind of the benchmark normal is 120/80. This top one is your systolic. The bottom one is diastolic. And this systolic, this is your ventricles contracting. And your diastolic are the atria contracting. Okay, and let's annotate this a little bit. So this, you know, just say that's your body. And this is to your lungs. You got two lungs. You got your two pathways here. And then it's going to come back from the lungs into the atria and then go back out. Okay? So that lower pressure is your diastolic, but you still have some pressure there. Okay? And when they, you know, look at your blood pressure, typically that top number is what you're focused on, but that bottom number is important too. You don't want that too high. If it's too high, that means your blood vessels aren't elastic enough, or a little too hard. So anyway, a lot can be determined from, you know, those blood pressure measurements. You've all had your blood pressure taken before, right? Okay. Kind of hurts, doesn't it? Squeeze that down. Get kind of nervous. That machine's going, or that person's all looking at that. Anyway, and we did the blood pressure in here. Okay, so at this time, what I want to do is give you guys time to work on those IA. I noticed only 2 people finished the one I assigned before the break. So, and I just opened up another one on data analysis. You know, are you going to use t-test, ANOVA, descriptive statistics? Well, everybody will do that. High five. High five for ANOVA. Okay, and also if you're at an impasse with your IA, there's also some videos and quizzes I put up there too for option D. We're almost done with option D. Okay, all right. So if you need a computer, I got some back there and up here if you want the other ones. Okay, all right. Any questions? All right, let's get to work on your IA documents or the videos. Instruction papers. Well, anybody in the class, yeah. So yeah, you should be thinking of something you can do. Notes on the internal, the IA. Oh yeah, the rubric. Yeah, I'm gonna have to dig around for that. I'm not sure if I could find any more of those around. maybe work on the videos, but there's also, you know, the documents, the IA documents that, you know, take you step by step through the process. That, you know, and the websites to explore, you know, what you can do. You can explore those too. You're welcome. On what disease? Um, mhm. Maybe you could look around for some online Alzheimer's simulation where you could plug in different variables. Potentially, that'd be a little tricky, but you could explore to see if there's anything that would be, you know, appropriate. Hmm. Hmm. If it's some kind of simulation where you plug in variables and you can generate your own data, like new data, it's not really meant to be like a report on something. It should be something where you're driving, you got a research question, and you can change some variables and look at data that's been published and come to a new conclusion, a new analysis. It can be tricky, but it's possible. You're welcome. Okay, so mung beans. Oh yeah, you're going to use bleach as an antibiotic. There's antibacterial. You kind of see how that kind of eliminating some of the soil bacteria affects the growth of the mung bean kind of idea. Okay. All right. So maybe some of your background research should be the importance. Oh, look at what you got. Is that your some new tech you're using now? You bringing your keyboard? Oh, you broke your laptop. Okay. Yeah, well, you adjust. So you could do some research on the importance of soil bacteria and microbes and how they are symbiosis, you know, how they help help plants grow. I think primarily bacteria in the soil help to, you know, break down nutrients, make nutrients more available for roots. But there could be other interactions too. And were you gonna do? Because we were trying to figure out how to apply the bleach or what to do with the bleach. Were you gonna? Were we thinking of using different tanks, like three different Lee Valley trays, and that way just you could add a certain amount of bleach to the water that they're being watered with, and then you could have like 3 levels of bleach. Have you looked at what levels are typically used? And then you can tie it in with your business, right? Is your personal interest statement? Yeah, I don't have it written down. I'm gonna write down 3 Lee Valley trays. It'd be nice to have more than 3 levels. So, I mean, we might have enough, trays. That'd be a lot of measuring too. That might be getting impractical. So I just don't know, like measuring, or adding the bleach to the single tray. I'm just worried about it soaking in and then contaminating or increasing the concentration of the ones nearby. But remember, you can't control everything. Yeah, exactly. And that's, and that's part of the experimentation process too, is just writing about those things. Because, yeah, so for this whole thing to be one level, you need probably at least 3 of these to have, you probably want 3 levels. But then you probably want one, you need one with nothing. So that'd be only 2 levels of bleach. But if you get, but the one that has no bleach should be independent at the same time. You shouldn't like divide this up. No bleach, 1 drop, and then another one of these will be 2 drops and 3 drops. Because, if you're adding bleach to this side, probably this side could get bleach, transmitted through the water somehow. So it's tricky. Yeah. Okay. Can you think of other ways to add the bleach besides, well, in a liquid form, they do make solid, like, aren't like bleach tablets or something? Or there's powder. Is there powder bleach? But regardless, it's going to be dissolved for it to get absorbed from the plant. So that's probably, just starting with liquid bleach is probably the easier way to go anyway. So, yeah. Did I mention, what did I say? I suggested mung beans. And you could put at least 2 in each of these, maybe 3. And then think about how many you'd have. Maybe you should just get 4 of these. Have 1. So you think about that. Okay. I need to get it all set up so I can see how many. I'm going to put, 3 or 4, Lee Valley. Okay. And Marcos, you were going to grind up Tylenol? You're going to grind up Tylenol? Oh, yeah. That's what I meant. So you're in the same similar situation. Marco's doing salt. So you gotta start grinding up some Tylenol. Have you looked at any other experiments and what concentration they use for maybe adding Tylenol to plants and see what? Have you looked that up? Plant experiments with Tylenol. The effects of Tylenol on plant growth. You should look into that. The World Wide Web has a lot of good stuff out there. But Marcos, your salt water thing, if you heard what I was talking to Aries Beth, you're kind of the same situation, right? You're treating using different salt levels. So you want to make it so that each group of plant is only being affected or influenced by that same salt level. We could use separate pots too. That's an idea also. Do you have something? What does it say? You gotta figure out, you're gonna grind up. You're gonna grind up Tylenol, and I don't know how well Tylenol mixes with water. I can't remember. It might not mix all that readily. How miscible with water it is. Probably you want the hard pills. Gel caps. Look at that gel stuff, and what does it have? Little tiny little spheres, little kind of grain things in there. Because I think. Yeah, I think I have. What? Let me see. Huh?
They have like a flavor to them. You put them in water and you mix them together.
Okay. Oh. That would be great.
I can use this like instead of
That's on the market now?
yeah.
Yeah, that would save you. That would save you. Yeah. Dissolve packs. So then you're looking at taking that dissolve pack, making a whatever the recommended solution is, and then you can maybe create different concentrations. Add twice as much water, a third more water, just to mess with the concentration. So you can change that variable and see if it affects the growth. Let's see. And Kenny, you have any sand available? Okay, well, let's see. I used to have sand. I'm not sure what's happened to it. I'll go look in the back. I think it might be in the back spot. And Jennifer, you're looking at plant light. So you just figure out how you're going to adjust the amount of the strength of the light, the filter or distance. Okay. What are you going to use for filter? You can use a screen, just a regular, bug screen, a window bug screen, because that would shade it, right? You could just do one with no shade, one with the shade, and see what happens. You could do even another one where you kind of put some holes in the shade or something. Okay. So we need to get you the PTC paper.
Yeah.
Okay. Have you filled out the other documents? Because basically you'll just have to start testing people and see if they taste. And you were gonna compare males and females?
Um,
Or what was your independent variable question going to be?
well, I think I was gonna touch everybody. Like,
Okay. Oh, and then, you can test. I think there's a statistics we could use, like chi-square, to see if we're different than the normal ratios. So did you look at the normal ratios for teenagers and their PTC testing, or a general population? So why don't you say what percent of people are PTC tasters? In the United States or maybe California. I'm not sure if that kind of data exists, but at least like in a general population, that should be able to find out what percent, and then we can compare our percent to that and see if we're statistically different. But the other idea is to try to break it another category too, like I said, males and females. See if there is a difference between males and females with the PTC tasting. Do males or females do they vary with being able to, do men tend to taste it more than women, or vice versa? So see if you can, that's something you want to put in your background anyway. You were going to look at adding coffee, okay? So do you have like a protocol ready to go and like a data table? So to start making like how are you going to write down the data? Because you're going to be using one of those Lee Valley. Have you looked at those seed starter trays? So you'll use one of those, or maybe two, and just have maybe regular soil, maybe all coffee, and then maybe half and half. Or maybe instead of all coffee, do like 1/3 coffee, half coffee, 2/3 coffee. Vary the amount of coffee. You're like putting coffee grounds in there. Okay. So think about how you're going to measure that out and adjust it. Yes, Madeline.
you know how doing like the difference in answering in your dominant and non-dominant?
Yeah.
So right here it says one Benny preliminary calculation for typical for distribution. Can I put the 10% rule? So that's like the 10% rule states that supposedly your dominant is 10% more stronger than your non-dominant. Maybe, right? Or no?
Yeah, you can look at. That's kind of different than your research question. What's your research question?
Is there a significant difference between the grip strength and dominant and non-dominant?
Okay, so your research says there is, right?
Yeah. So
you're just kind of testing that.
Yeah. But
Okay.
it's like a not theory, it's just a fact that
So you can test people's they say it's 10%?
Yeah.
Okay.
10% rule states that dominant has a 10% greater grip strength than the non-dominant.
Okay. And that's from where? The NIH?
Yeah.
Oh, okay. So that's a pretty good. See if you can have that source and quote it and write about it more in your introduction, and definitely cite that. See if you can find some other sources too. Like they probably got that from many sources. You can't have a rule without lots of different, people deciding on it. Yes. You're not feeling your IA? Justice system. Oh, digest. I was like, you were going to do spoilage, right? Or no? Yeah. you're yeah, you were just going to look at the rates of how the pH changed over time of the milk as it spoiled. Is that what you were going to do? Say that again. Different kind of spoilage, or what? What's spoilage? Were you going to use different types of milk? Okay. Yeah. Those are really different things because milk has bacteria in it, just low levels, right? Unless it spoils. Almond milk won't have any of that because it's not a dairy product. It's just crushed almonds and water filtered through crushed almonds. It'll have some fats and protein and stuff like that, like milk has. So it has kind of that feel to it. But like the spoilage, for your angle is digestion. Did you look up IB biology IAs on digestion? Or just anything? You just search IB biology IAs on digestion. IB milk spoilage. One came up? And what was its procedure? Different light colors. So look at that. Oh, there we go. Did they wrap clear bottles of milk in different color, plastic? Yeah, but like how they make the lights different colors. One way to do that would be to wrap the bottle like in a film, like a colored saran wrap, and then put it under light, and then that light will turn into that, be filtered.
The LED light.
Oh, they use LED and they change the color. I don't think how do we do that? How do they do it? Where do they get the LED light, like a LED flashlight? Yeah. How stomach acids and enzymes affect the milk. So how can you? Well, lactose is the sugar in milk. Lacto. Well, people make lactase, which is an enzyme that breaks down lactose, and I'm not sure. I imagine that comes out in the small intestine. I don't know though. You could look into it. Where is the lactose digested? Probably because it's a sugar. you digest some break down some proteins in the stomach, but sugars are digested either in the mouth or then in the small intestine. So I imagine the lactase comes out, the pancreas probably releases it into the small intestine. You're talking about the large intestine or something?
Something about how like the large intestine, if it doesn't process something like if it like it goes in the small intestine and if it doesn't process it or something like that, it goes it like transfers to the large intestine, which is when you get like the bloating.
Oh yeah, if it's not digested, yeah, that's where the bacteria in your large intestine start to eat it up. You don't really have a lot of bacteria, if any, in your, I don't know, there's probably some in the small intestine, but it's when the large intestine bacteria start to eat it, and then people get sick, right? Gassy, diarrhea, that kind of stuff, because of the bacteria eating it. So you want to do some experiment looking at that stuff. So I wonder if, so you could put milk in different bottles, and you could measure the pressure. We have gas pressure things you could use to measure the pressure building up inside. That's one way. Another way could be, using pH, because the more the bacteria, as the bacteria eat up the lactose, does that change the pH? Does that make it higher or lower? Or does it not change it? I'm not sure about that. What are the... Yeah, and I don't... we can't, like the bacteria in your gut, we can't, I don't know how we'd replicate it. We can't, grow any bacteria that can be dangerous, right? Like E. coli, that's in your large intestine, but we can't really grow E. coli safely, really. I mean, we could, but probably want to avoid it. But I'm wondering if just like letting it sit. I mean, what about, you know how you get those tablets? You can buy those tablets. Yeah, like people who are lactose intolerant can use those to eat them beforehand, and that helps to break down the milk. I wonder if you do some experiment with those tablets. No, not on yourself, but you set up different bottles with the milk and have different amounts of the tablet powder in there. I might have some in my stuff, actually. I think somebody messed with those before. Why don't you look at that? IB, IA biology experiments using Lactaid tablets. Because it's supposed to break down, it should break down the sugars. So if we could figure out a way to measure the sugars, what you could also do is you could like heat treat the milk or do different things in the milk and then see if that affects how well the Lactaid works.
Like warming it up or like heating it up.
Yeah, you can do simple that as microwave for different amounts of time. This milk wasn't microwave. This milk was microwave for 10 seconds. This for 20 seconds. This for 30 seconds. 45, a minute. Does that increased level of microwaving affect the outcome of the experiment? Does it change the milk in a way that affects the action of the Lactaid tablet? Or you could add, different amounts of acid to the milk and see if that affects how the lactate works. Because, supposedly, your own enzyme that's not working, it's not there when you have lactose intolerance. And what affects the action of enzymes? Acids. Heat. Yeah, acids and heat are the main things that affect the shape of the enzyme and how it works. So that could be an angle too. Having different milks, adding different levels of acid, and then adding lactate, like the same amount of lactate to each one, and then the trick is measuring its action. Like I said, we could try to measure the pressure. We can measure maybe the pH. But looking at IB biology, IA experiments with lactase. Let me write it. Oh, so lactase. Like I said, I think they might be kind of old, but I thought I had some tablets in one of those boxes over there. Well, it depends on what your research question is. If you're looking at different milk, now remember, almond milk's not going to behave the same way at all. Or what are the other? Coke, soy milk? Yeah, they're not dairy products. They're not, because milk comes out with a certain amount of bacteria in it. So what they do is they treat it to knock down the level. They knock it down. There's still some bacteria in there, though. Yeah. Not sure. Like in Europe, they get rid of all of the bacteria from the milk, but we don't here. I can't remember why that is. And I don't know the exact health consequences either. Yeah, I think there is a benefit, but I'm not quite sure. Same season's been on the whole time.